sometimes in evolution, some parallels appear pretty quickly. Yeah. There's an awful lot of things that you study in patients has direct relevance for evolutionary studies as well. So polyploidy. Well, polyploidy arises as a result of dispermine embryos, we know that. Translocations can cause infertility in an individual, but they can also cause a reproductive barrier, such the hybrids are pretty much the same as someone who would have a heterozygous translocation. Same for inversions. Now, we don't get much aneuploidy in evolutionary studies, but we get a lot of fissions and fusions, insertions, deletions, and CMVs, and so on. And, well, if you want to understand biology better, then looking in the context of evolution is never such a bad thing. So what do we do in practice? Well, I'll just use 3 birds here. This is from a paper that we published in 2014. Chicken versus turkey versus zebrafinch, back versus turkey again. And the blue bits means that the sequences are in orientation, whereas the red ones, as I said before, represent an inversion. When we study these things, we look for 2 things. The first are the homologous synteny blocks, and the second, the evolutionary breakpoint regions. So in this diagram, the HSBs are represented by these colored arrows that are sort of in rainbow colors, and those are delineated by the EBRs, the evolutionary breakpoint regions. And I don't have enough time, but if I had enough time, I would start thinking about telling you about how we can look at the gene ontology of these regions and how it relates to phenotype. But for the purposes of this talk, one of the things it allows us to do when we have 3 species, or at least more than 2, is get some idea of the direction of change. So this is chromosome 9, and this is our ancestor of chicken, turkey, and zebrafinch. And we think, for instance, there's been just 1 change in the chicken, but several in the zebrafinch. How do we do that? What's the principle? Right. If we consider chromosome 2 of a human, this is a human, that's his chromosome 2, like so, and compare it to that of his nearest relative, our chimpanzee. Now, disclosure here, this is me messing about with Adobe Photoshop, okay? The chimpanzee chromosome looks almost like that, but you'll see that really all I've done is chop them in half like so, okay? But it makes the point. Now, the question that we want to ask ourselves in evolutionary studies is, what did the ancestor have? Okay. So what came first? So did the ancestor have the human-like pattern, and was there a fission? Or did the ancestor have a chimp-like pattern, and was there a fusion? Now, obviously, the one thing we can't do is go back 6 million years and take a blood sample from the colony of proto-humans and chimps. But what you can do is start comparing the next most related species, what we call outgroups. And then when we do that, we discover that the gorilla has the same pattern as a chimp, as does the orangutan. So if we look at the relationships between them, then we can say with confidence that the event was a fusion that happened in our lineage. So the chromosome 2 that you see when you see a karyotype is the mark of a human, and only a human. Okay, similar thing in birds. Chicken, turkey, chromosome 2. Okay? Right, same thing, yeah? Well, actually no, because if you look at a goose or an emu, you see that they have the pattern that's similar to the chicken. So in this case, it's a fission. Another analogy for you. This is my oldest son a few years ago. Take half a second to describe what you see. Boy standing on a crack in a rock, yeah? Well, actually, no. This is Iceland. His left foot is in North America, and his right foot is in Eurasia. So he's actually standing on a fusion, not a fission in that case. So where do we go with this? Well, we needed to start comparing more species. This is Mike Romanoff, who works in my lab. And at this point, we were up to budgies and zebrafinches and ostrich, as well as chickens, turkeys, and ducks as well. And these are just some of the HSBs in the different species and their orientation. As a result of these studies, and by using our outgroup of our green anole lizard, then we were able to determine what we think the ancestor looked like. Now, in the example I gave you, you can just do it by hand. It's very easy. But once you start looking at multiple species, it's best to start using some bioinformatics tools. And the one that we used here was MGRA, or the Multiple Genome Rearrangement and Analysis tool. And this is just an example of one of the chromosomes. This is chromosome 11. And we have our avian ancestor up here. The changes that have gone on to form our zebrafinch, our budgerigar, our turkey, and our duck. And we see the ostrich and chicken were pretty much the same as the ancestor. So what about this ancestor then? What was it? What was this creature? Well, I talked to some of my friends at the Natural History Museum, and they said to me, well, probably bipedal, probably feathered, and probably about the size of a chicken was the answer. What it was, in effect, was a 150 million year old Jurassic dinosaur. So we thought, oh, well, hang on a minute. Maybe then we could start looking at some dinosaur lineages generally. Now, I have to say, it is fun to work on dinosaurs. They have captured the imagination of scientists, the creative arts, the general public since those very early fossil discoveries. And as a result of the efforts of Michael Crichton and Steven Spielberg, we suddenly got interested in their genomics as well, and with that sort of iconic image of Richard Attenborough there. So what happened to them? Well, they're all extinct, yeah? Hold that thought. Well, there was an extinction event Permian-Triassic, massive volcano eruptions 250 years ago, and that paved the way for them to appear at about 245 to 240 million years ago. 228 million years ago, they survived one extinction event, as they did 201 million years ago, that wiped out the crocs, all but 23 species, and something like there are about 1,000 dinosaur species now described, with about 30 more identified each year. The extinction event that you've probably heard about most is a Cyclope impactor meteor strike that caused an extreme reduction in temperature and daylight, and it wiped out most of the dinosaurs, but not all of them. There was one theropod lineage that survived, and that emerged again as the modern birds. So if you wanted to look down the line of where things, how things are characterized, we start with diapsids up here. We have archelosaurs, which are basically birds and turtles. The turtles disappeared off at about 260 million years ago. About 240 million years ago, off went the crocs, leaving the pterosaurs and the dinosaurs. Off went the pterosaurs, and then we had this theropod lineage all the way down to modern birds. So birds are dinosaurs, pure and simple. Birds did not evolve from dinosaurs. They're not related to dinosaurs. Birds are dinosaurs. Okay. So here's some of the questions that we started asking ourselves. Can we reconstruct ancestral dinosaur genomes by inference? Did they have bird-like karyotypes? And does this explain, in part, their biodiversity and resistance to climate change? So let's have a think. How did that work? So what we did was we adapted our bioinformatic and fish-based approach, and we started looking with our highly conserved probes on some reptile species, including a couple of turtles. And one of the things, even just by looking at a regular karyotype that jumped out at us, was there are some turtles, not all, but some turtles that have very, very bird-looking karyotypes. Birds have loads and loads of chromosomes, loads of tiny ones like so. But just a handful of turtles seem to have something similar. And when we started looking more closely, one of the things we started finding was not only do they look the same, but they are synthetic to one another. So chromosome 1 in this turtle is a 1 in a chicken, and a 2 is a 3, and so on. And here's just some of the examples of some of the tiny chromosomes before they split off. Now, couldn't we have looked at crocodiles? They're more closely related. Well, yes, we could, but unfortunately, chromosomally, they fused all their chromosomes back together again. So crocs are bad for this particular exercise, but we have been doing some stuff. But here's the bottom line for our studies on dinosaurs. Our diapsid ancestor about 270 million years ago had classic lizard-looking karyotype. It rearranged to this sort of turtle-stroke-bird-like about 260 million years ago. It underwent a few more fissions. Remember, there's another 20 million years before the dinosaurs appeared. So we think that this karyotype that you see in a chicken is pretty much found its way into the dinosaur lineage about the time the dinosaurs appeared. And it remained fixed, which is pretty stunning. So, this weird karyotype, why? Well, we've combined lots of aspects of cytogenetics and bioinformatics. We think that it became fixed around 240 million years ago. Why would that be? Well, either no opportunity for change or an evolutionary advantage to keep it that way. Well, the genomes of birds are small. We think that that applied to theropod dinosaurs as well. So your opportunity for change is reduced, but you can split. You can still undergo fission with that. But lots of chromosomes means you can shuffle your genome a lot by random segregation and by recombination as well. So with this increased recombination rate and random segregation, we think that both the configuration is evolutionarily successful and there's an absence for positive change. So that's why it's stuck around. But importantly, what it's allowed us to do is not only just look at this avian ancestor, but also the lineage of dinosaurs. And obviously we don't know how many dinosaurs had this karyotype, but because they have survived, and when they get wiped out, because of all this biodiversity, biodiversity is the driver of natural selection, perhaps as a result of this funny-looking set of chromosomes, that's what allows them to go on and survive. And if you want to have a look at our paper, it was published earlier this year in Nature Communications. So we think this not only leads to this, our variation in birds, but perhaps to this as well, or at least is a contributing factor. But the question, of course, I get asked most is, does this mean that this will be a real Jurassic Park? And there's Becky being interviewed in my lab by the BBC. Well, we wrote a paper on this. It was published in Scientific American this year as well. Occasionally, when you say something to the press, odd things come out like that. So apparently we're trying to clone a dodo. That was news to me, but there you go. But basically, the crux of this article was, first of all, the DNA won't survive that long. This is actually a well-established field. We've got Neanderthal men and woolly mammoth DNA. Cave bears are particularly well-studied, but you can get an idea of how ancient DNA degrades, and it's a long time before 66 million years. It would be fragmented, unclear of organization and gaps. We can't just fill it in with frog DNA like they did in Jurassic Park, and realistically, developmental biology doesn't work like that. How on earth do you recreate that egg with all those biochemical clues to get the genes activating on and off at the same time? Theoretically, you could resurrect, say, carrier pigeons that only went extinct 100 years ago using that approach. That might be more reasonable. Evo-devo studies, gene function studies looking at dino genes by manipulating chick embryos. That is a very interesting area, but no, certainly no Jurassic Park. So I'll leave with some parting thoughts now. Basically, we talked to you about these extinction events. The birds survived, but let's get a reality check here. We're living in an extinction event. Something like since 1970, I was reading, about 60% of species have been wiped out. But stupidly, we caused it. And the birds, perhaps because of their chromosomes, might just survive it, but we might not. So I'll end with this. For the first time in 66 million years, karyotype of Velociraptor looked probably something like that. And that just leaves me to thank all the folks that I've worked with, including the guys in my lab. Thank you very much.